So welcome everyone. I'd like to welcome you to our fourth installment of Proteoform Thursdays, our new monthly seminar series. I'm Paul Danis with the consortium, and I'll be hosting today. And we're fortunate to have Charlotte Uetrecht with us, and she's going to be speaking about some of her fascinating new work on native MS and the structure of viruses. So let me just give you a quick blurb on the consortium. The main things we do is we look around building the community through education, conferences, workshops, and we also try to help guide the technology around developing protocols, doing community assessments and things like that, as well as building the proteoform database. So one thing we did recently when folks registered was we asked kind of what's the main challenge you have, and this was kind of interesting to see. We didn't know what to expect. We knew there were lots of challenges, but the sample preparation was above the other ones as far as the biggest challenges. But then across the board, you have sensitivity, data analysis, coverage and databases, kind of all 10 to 15% of the issues. So the challenges are spread across the board here. It's not like there's one thing that everyone's wrestling with. So it's on one hand, it's lots of opportunities for research and development to improve all these areas. The other is it's really hard to do this because there's lots of places people can get caught up. So one thing we've done is we've developed a course that's available this fall. It's inaugural rollout. Ing, Neil, Joe, and Lily will be the instructors in this course. And it should be fascinating. It's people that are new into the area or looking to get into it or want to brush up on a few areas. It's going to deal with sample preparation and separation, instrumentation methods, looking at top down versus bottom up, what are the pluses and minuses of each, the whole data interpretation thing, quantitation of proteoforms, and then applications in the biopharmaceutical world. So if you're interested, take a look at asms.org. They have this available for sign up, and that should be fascinating. Next month, we have Kyle Brown from Wisconsin. He's going to be speaking about membrane protein work. He's been doing that, and there'll be more details coming out over the next week or so on that. And if you're not involved yet as a member of the community, it's free to join the consortium. You can just go to the website, either topdownproteomics.org or just type proteoforms.org. That'll get you there. And happy to have you join and be engaged more with the community. So finally, I'd like to thank our consortium sponsors, Thermo Fisher, Bruker, and SciEx for the generous support, as well as support from Agilent and Pfizer. So with that, let me turn it over to Charlotte, who is currently at the Leibniz Institute for Experimental Virology, and will also be getting a position at the University of Siegen shortly. She'll be speaking to us about flying viruses from biophysical to structural characterization. So take it away, Charlotte.
Thanks, Paul, for the kind introduction, and also for inviting me, of course, because what we do is not really proteomics. It's certainly top-down. We mainly do native top-down to investigate virus structure, and this is a little teaser that the Leibniz organization recorded for the Leibniz year, and I think it nicely illustrates that viruses fly. Last but not least, from one host to the other, and the current pandemic has, I think, made us all aware of that. And I usually use that as an argument to say, so why not analyze them in the gas phase if they are happy flying around anyhow, right? So just a little recap for the people here what native MS is. So I guess most people in the top-down field work denatured. So you put your originally nicely folded protein, which is the VP1 from noroviruses here. We will get back to that later on. If you stick that in acidified organic solvent, what you will get is a more or less unfolded protein chain that is then extended and can take up a lot of charges. And the type of electrospray mass spectrum you then get will feature a lot of different charge states over a broad M/Z range, reflecting the unfolded nature of the protein, where you have compact states with a low charge and then very unfolded states with the high charges. What is also evident here is that our VP1 here comes in 2 flavors, A and B, which are due to different methionine processing, and the ratio is, well, not exactly equal, but both are quite abundant. If we now take the very same protein and do not put it into organic solvent, but into buffered solution, or I should better say into our buffer surrogate, ammonium acetate, and we spray at neutral pH, our M/Z changes by a factor of 10, and we no longer have the monomers, but we have an intact 180 mer, which is the protein shell that normally protects the genome. And this whole thing has 10 megadalton in mass. We no longer have the charge states here because of the different forms A and B, which come in multiple mixed ratios. And in the low mass range, we now see a VP1 dimer, which further indicates that we are looking at folded protein, because the dimer is the building block of this whole protein shell. If you have protein without microheterogeneity, like is the case for hepatitis B virus capsids, then you can still get charge state resolution even on a very old QTOF, where this data is originating from, and you can very precisely determine the mass. As I said, this is QTOF data. We haven't tested, but I would guess that exactly this sample with this very strong microheterogeneity would also be a challenge for an Uetrecht UHMR. I'll tell you once we've measured that guy. So what kind of information can we get from native MS? Since we measure mass, we can of course say something about the stoichiometry in a protein complex, which is one of the main questions oftentimes. How do my subunits assemble? In combination with dissociation in solution and gas phase dissociation, we mainly do CID. We can also say something about topology, and I will have an example today for that. In combination with ion mobility, we can also say something about shape and conformational change. And since the signal is oftentimes very well related to the concentration in solution, we can also say something about the ratios of these different states that are present. Since the ratios are a good reflection of the solution concentrations, we can also study binding equilibria. We can also say something about the direct number of ligands bound to a protein, which is not easily accessible by other techniques. We can provide KDs. Of course, I don't have to tell you that we have to take care of artifacts arising during the electrospray process, but for many ligands, this works very nicely. And then, of course, we can follow the mass evolution over time and thereby monitor protein dynamics. And to me, this is the most interesting thing, because when you talk to the structural biologists or virologists, there are a lot of things they have in their toolbox to look at the structure at high resolution. And of course, we cannot provide that. But what they often cannot do is follow the dynamics. But we can, and we can do that with good time resolution and with very high mass sensitivity. And so a while ago, even before the pandemic, we set out to use native MS and understand coronaviral replication transcription complexes. You're, of course, by now all coronavirus experts, but until the early 2000s, coronaviruses were known to cause mild cold in humans. For example, human coronavirus 229E, and nothing severe. So there was not a lot of funding going into research on human coronaviruses. It was considered boring. And then SARS came around and everybody started working on coronavirus. Then SARS disappeared, funding stopped, MERS came, people started working on coronaviruses again, funding cranked up, and then, you know, it kind of drizzled down again, and now COVID-19, and I guess you know that there's enormous amounts of money flowing into that. So there are these severe zoonoses. For those of you not familiar with the term, zoonosis means a virus or pathogen in general moving from one host to another, from an animal to humans, for example. But for a long time, coronaviruses were known as severe disease-causing agents in animals. For example, in cats, where we have the PIP virus causes severe disease. Many cats die of that. Then we have two porcine coronaviruses, TGF and PEDV, which cause gastroenteritis, and they have caused severe losses in the pork industry. They affect mainly piglets. So the replication machinery is more or less common to all coronaviruses. It's produced from a huge polyprotein that is expressed in the host cell, and then two viral proteases chop this polyprotein into smaller pieces, 16 non-structural proteins that can be further modified, as exemplified here by the P for phosphorylations, and that then assemble in or around, that's still debated, these double membrane vesicles into the replication transcription complex, and then generate new genome mRNAs and all what's needed to maintain the infection. So one thing we started out actually last summer is joining a collaboration in Hamburg to assist screening of already approved drugs against the main viral protease, MPRO. And here you see some examples. This is the native mass spectrum of the protease. We see a little bit of monomer and the dimer here. The dimer is the active form, so that's what we're looking for. And if we zoom in on the main peak, we can see that this drug candidate actually binds pretty efficiently to the protease. It occupies both active sites at relatively low input concentration. Similarly for calpeptine, which also other people have identified as a good binder. And then we have some not as strong binders, like this guy here. You can see there's still free protein around. And then we have this heat, which is also not so effective, but it is interesting, oops, wrong direction, because it's actually a covalent bond that is created here. So we have an irreversible inactivation of the protease. And we can see that because the compound is still bound to the monomer. When we release that in CID, you can see the mass shift here of 159 Dalton. And this is further confirmed because the mass is reduced compared to the input compound. So there's definitely some chemical conversion going on. But what we were really interested in is this polyprotein processing that I've talked about earlier. And here you see a structure of the polyprotein that comes in two flavors, short version and a longer version. Towards the front end, and I define the front end up to here, NSP6, NSP for non-structural protein. We find the membrane proteins and some other effectors in the cell, but also the main protease here, NSP5. And then we have NSP7 to 10 or 11 following, which are modulators of the main enzymes in the replication transcription complex, which you only find in the long version of the polyprotein. And I think by now, for all of these enzymes, there have been critical interactions identified with these small guys here in the 7 to 10 stretch. And it's also known that this stretch is existing as a polyprotein intermediate in infected cells. So we wanted to know, can we use native mass spectrometry to follow the processing of this stretch by the main protease? And this actually made it to the cover early last year in Biochemical Journal, where you see the polyprotein processing and assembly. So what did we get? This is just an example. If you take this NSP 7 to 10 stretch and incubate it with protease for a couple of hours, you get these very complicated spectra. On the right-hand side, we have the protease dimer, we have our substrate, the polyprotein here. We also have some complexes that form after processing. I will get back to that later. We see intermediate species, and we see the free monomers and homodimers, basically, in the low MOVZ. Since this is very difficult to look at, we created some easy plots. So what you see over time, and time is here 20 hours, you see quickly the substrate intensity goes down, while the protease level stays constant, which tells us there's no massive aggregation ongoing, and there's also no self-digest of the protease. It's a very specific protease. There should be no self-digest. If we look at the monomers, we see they almost all end up at the same mass fraction in the end. So we have mostly complete processing, and we see that NSP10 and NSP9 come up very early, and NSP7 and 8 are released with delay. This is further corroborated if we look at the intermediate species. So we see NSP7-9 coming up first, and then with a delay, NSP7-8 is peaking, and it stays around for a long, long time. So from that, we can determine the processing order. So we start from the C-terminus and then release the individual proteins. This may sound boring, but you have to keep in mind that all these little colored bars are folded protein domains. So the protease is not walking along it. It has to access each side separately. And there's certainly some functional aspect why NSP7 and 8 are released later. And another thing that we did back then is we tested the processing speed in peptide substrates, just covering this protease active region. And then we had these labeled with fluorophores, so we could monitor when the peptide is cleaved. And if we do that, we have a positive control here that's super fast. If we do that, we see that the cleavage site 9-10 is fastest, as we had seen in native MS. But the second we see is 7-8, which was slowest in native, and 8-9 is virtually dead. So this tells us that structural context here is important. Only if we have the folded domains around, we get the correct processing order. And the reason why this is dead is probably because there is peptide secondary structure if you have these short constructs, which is not the case if you have the folded proteins around. Okay, another thing we were interested in is the 7-8 protein complexes that form after processing. And we have looked at that for many coronaviruses. So what you see here is SARS-CoV at the top and SARS-CoV-2 at the bottom. What we see in the spectra are 7 and 8 heterotetramers, so 2 times 7, 2 times 8 coming together. We also see the protease because we freshly cleaved the protein to make sure there is no decay whatsoever. We also see heterodimer, which is probably an assembly intermediate. We see the free monomers. We see a little bit of NSP8 dimer. And here you see the deconvoluted spectra, which are probably easier to look at. So from this, we can clearly say that these SARS coronaviruses both form heterotetramers for the NSP7-8, which is the main cofactor for the viral polymerase. We also looked at alpha coronaviruses. This is the cat virus I mentioned before, and this is one of the pig viruses. And here we get a different picture. We don't have a heterotetramer, but we have a heterotrimer with 2 copies of NSP7 and a single NSP8. Again, we see the heterodimer as an intermediate, but nothing else. So from here on, we could have stopped and concluded beta coronaviruses form tetramers and alpha coronaviruses form trimers. But we happen to have more viruses or viral constructs around. And it turned out that some alpha and beta coronaviruses can form both trimer and tetramer. You see that here for the common cold human coronavirus, or maybe better in the inset, we have the trimer with 2 NSP7, and we have the tetramer with 2 copies of each. And the same for the porcine virus, and also the same for MERS coronavirus. This is a bit more complicated because we had to use a different construct, but also here we get the trimer and the tetramer, and additional intermediates that we hadn't seen in the processing for the SARS-CoV NSP7 to 10 stretch. So that's also something we're looking more into. So the question now is, if we have these trimers and tetramers, what do they, how do they look? And we went into the PDB, and these are the full length structures you find for the 2 proteins. There is a structure for SARS-CoV NSP7 and 8, which is an 8x8 heterohexadecamer. And if we extract from that tetramers, what we get are these 2 types that are possible interacting via NSP8. And then there is a crystal structure for the cat virus, where we actually see a trimer like we saw in native MS with 2 NSP7 that directly interact. Note, some of you may have seen that there are SARS-CoV-2 NSP7 and 8 structures in the PDB, but none of these is full length. They all contain truncated NSP8, and what is truncated is the interaction region that is seen in SARS-CoV-2. So that might be an artificial interaction. I just want to point that out. That's why I'm not showing them here. So, but what we of course wanted to know, why don't we see this 8x8 complex? So native MS only works at low concentrations. So we went to some other technique that gives us higher concentrations, which is dynamic light scattering. You see that here for 2 different concentrations, and within the error, the radius obtained is the same. And this is also true for SAX, small angle X-ray scattering experiments, where the curves do not change a lot while we go to higher concentrations. And most importantly, they fit a tetrameric structure, but there's clearly nothing that fits this hexadecamer, even at 30 mg per mil, and the relevant concentration, or theoretically relevant concentration maximum, is somewhere here at 15 mg per mil. Okay, so we have this trimer and tetramer. The next question is, do these look the same? Are there differences? What about the topology? So as an example, I'm only showing the human coronavirus here. We selected the tetramer in CID. And if we do that, here's the precursor, and gradually crank up the voltage, we get consecutive subunit losses. And important is we see 2 types of dimers. One is the heterodimer of NSP7 and 8 here, that we have also seen as assembly intermediate in the native MS. And we see an NSP8 dimer. So from this, we concluded that we must have direct interaction between 7 and 8, and 8 and 8. So this is the topology map, which nicely fits the extracted structures from the SARS-CoV crystal structure. If we do the same thing on the trimer in the HCoV sample, we get a different picture. We get the heterodimer again, and we get an NSP7 dimer. So here we have NSP7 direct interaction and then association to 1 NSP8. And this is the same, we have the same topology for all trimers, and we have the same topology for all tetramers that we observe. So there are 2 distinct assembly pathways for NSP7 and 8, and some viruses are capable to follow both routes. Then, of course, the question is, why do they do that? What are the structural implications? So here are again the structures that I showed you before. And basically in all structures, we have the same binding sites on NSP8 being the main players, binding site 2 and binding site 1. You can see them highlighted here. And if we look at the atomistic detail, then we also see that the same residues are involved. For example, here this methionine pops up in almost all, and here the other methionine as well. So there seems to be not really a difference. But if you do a sequence alignment, there are actually very few differences. And you see all the viruses that we tested here and the group assignment. So B is trimer only, A is tetramer only, and this is both. And if we now look into the structure where these residues are, we have, for example, this area here. We see that the glutamate 77 self-interacts in the NSP8 interface in the SARS-CoV structure. And if we do a homology model for the human coronavirus, actually the asparagine can take on the job of the glutamate and do this interaction. And we think the aspartate in the trimer only can't do that because it's the only acidic residue and it's actually solvent exposed. So, okay, so much about the tetramer, but what about the trimer? If we have a close look there, we have a hydrophobic interaction here mediated by phenylalanine in NSP7 and leucine in NSP8, which you can see here. And that is only found in those proteins that can form the trimer. So the group AB indeed has both features, at least as we pinpointed that. And we're currently producing mutants to prove that this is indeed the molecular switch, if you wish. What is also interesting, if you look at the interaction with the viral polymerase, that's the thing here in beige, and the helicases here in blue, you see that the NSP8 uses the very same residues that are playing a role in self-interaction and interaction with the second NSP7. They also interact with the helicase, which probably explains why there's never a tetramer bound to the polymerase. But I could envision, because these residues are not interacting with the RNA, that maybe the heterotetramer could bring together two polymerases, which would be biologically interesting. Okay, a little bit to the noroviruses that I've mentioned as example for native MS in the very beginning. These cause viral gastroenteritis, and I'm pretty sure you've all heard about that. They are non-envelope viruses, which means the shell that is formed by the protein also directly mediates host cell interaction, and it encloses the single-stranded RNA genome. And usually it's forming, the protein is forming a T=3 capsid, which means it's containing 180 subunits. So the T number, you always have to multiply by 60 to get the subunit number. And already during my PhD in the HEC lab, we looked at the prototype norovirus, which is the G11 Norwalk, and investigated the pH stability. And what we observed back then is if we go to alkaline pH, we get conversion from the T3 to T1 particles, which contain 60 subunits. So a few years ago, we started revisiting that because we wanted to know whether different variants have different stabilities. And indeed, that was the case. So we had G11 Westchester, which was more sensitive to alkaline treatment, and G217 Kawasaki, which was less sensitive. And most importantly, both of them showed the T=1 particles already at neutral pH. And back then we hypothesized that this has to do with amino acid changes in the N-terminus. And then we went on looking at further variants, and some of them mainly did or exclusively did in this case T=1 particles, and we were wondering why that is. And if you do CID at these guys, you see consecutive monomer losses confirming that we have 60mer. But what you also get is mass that corresponds to truncated capsid protein. So when the N-terminus is lost and the switch is roughly 45 amino acids, then you get smaller particles only. or almost only. And together with people at the TU in Vienna, we also did gamma, and what we observed there, comparing a batch in green that contained the full length protein and the one with the truncated, doing T=1 only at alkaline pH, we could see that the truncated version is still stable. And in collaboration with Martin Gerald, we did CDMS, and what we could actually see is that there are interesting intermediate states that we had not expected, because they are not cannot be multiplied with 60, between 60 and 120 subunits, that point to possible different assembly pathways. And this leads me to the very last thing, and I promise I'll be short on that, Paul. But we are interested in how these other assemblies look, and also assembly intermediates prior to the full capsid. And this is one of the reasons why I moved to Hamburg some 10 years ago almost. And we have a very nice European project on that. And we just released the comic strip, which explains you what we're doing and why noroviruses are important to be imaged at high resolution. You can access that via this link. So what are we doing? In Hamburg, there is a huge free electron lasers providing hard X-rays called European XFEL. That's what you see up here. What it basically does is it brings electrons to super high speed, then sends that through an alternating magnet, so they release X-rays. And these X-rays are very high intensity and very short pulses. And due to the short pulses, you create diffraction patterns of your particle before the particle actually explodes. So you can do single particle imaging, but you have to record many, many images. And the problem is often sample heterogeneity, especially if we talk about intermediate states. So I came up with the idea to combine that with native mass spectrometry to clean up gas background, water background, separate mixtures using a quadrupole mass filter, ion trap to increase density, ion mobility for conformational separation. Yeah, this is the schematic outline of the instrument. We are even playing around with dipole orientation to ease the pattern alignment later on. And then we have a diagnostic TOF. So where are we in this project? We have all the modules for the prototype in hand, luckily now. And the ion transfer interface can transmit up to norovirus, we know that, and very efficiently. The digital filter and ion trap has been tested up to GROEL, which I think is pretty good as well. The ion mobility device from Purdy Barron has been tested mainly on standards now, but the real life testing will follow soon. And the dipole orientation has been investigated theoretically. And the high field device has been built. There's no sparking, but the real life testing with proteins is still pending, because we still have to put all these modules together and then test it at the European XFEL. And there have been shutdowns and what's not, so this got delayed. Okay, to sum up, stay tuned for native MS at European XFEL. Have a look at the comic. I showed you how top-down MS can provide insights into norovirus assembly, also only with a short glimpse, and then how we investigate non-structural proteins in coronaviruses, moving from inhibitor screening to the distinct complexation behavior. And of course, there's no way I can do that all myself, a little outdated group picture, but I certainly want to point out Boris, who did all the coronavirus work and is also in the audience, so I may pose some questions to him. This is the consortium for the XFEL stuff, lots of funding sources, and I thank you for your attention.
Great, thank you very much, Charlotte. Fascinating work, that's for sure. If you get a chance, could you post that URL for your cartoon into the chat so people can take a look at it?
Yeah, I can. Oops, I need to exit that one. Then I can do that.
Yeah, and if people have questions, feel free to unmute yourself and you can ask a question live. Or if you prefer, you could type it into the chat as well and we can address it there. I might just add a sort of more of a general sort of stepping back question. I mean, your work is pretty advanced and there's some folks probably who are looking to try to do this kind of work. So is there a simple system you could suggest that people could start with to try to develop their native work?
Well, the most simple system, you mean protein-wise or instrument-wise or both?
Yeah, protein-wise, and you can comment on the instrument thing, but more the protein-wise, what should people, where can they, get a toehold and start to develop their methods?
Easiest are small proteins, no surprise. Don't start out with viral proteins. They are tremendously challenging, except for the protease. Well-folded, stable proteins are best to start out with. If you can find from extremophiles is very nice. But you can even start off with proteins from Sigma. We use it as standards, for example, myoglobin, that carries a heme group non-covalently. So if you only see apomyoglobin from Sigma, then something is wrong, then your workflow is not native. So that's probably the easiest to start with. And oh, the link is not working. That's a pity. I'll paste it again in a second. Just get some standards that you can get in high quantity, get experience with your mass spec, and then take it from there.
Could I ask a follow-up to that?
Sure, go right ahead.
Yeah, what about people who already know how to do native and do a lot of different kinds of protein complexes with native, but they would like to get started on the viruses? Are there commercially available capsid proteins, for example, that will self-assemble that we could buy and use?
So for the capsids, I think there's some commercial source for adeno-associated virus. At least there are companies producing that as vectors. You can either get in touch with the companies directly, or check out whether they really have it online. I'm not 100% sure because we produce most of the stuff in-house, of course.
Thank you.
Other folks with questions they'd like to ask? Maybe you could comment on the instrument side as far as how to develop your system to do both, maybe initial native work and then maybe more advanced things.
So from my point of view, and that's what we usually start training students on, is just an easy tough system, because there's no way, or it's not so easy to damage. There's not a lot of things you need to tune. We have some of these old LCTs. Somebody found a correct link. Excellent. Because answering questions and looking for the link at the same time is a bit tedious. So yeah, these LCT systems, they're no longer built, but we use that a lot. And for ligand binding, that's very nice. The less settings you need to tune, that certainly helps. If you have to play with a lot of parameters without knowing which are the most critical ones, you have a hard time. Then you certainly should go to a lab that has the machine you intend to buy. We have a UHMR as well. It's a really great instrument where people love it, but get some training somewhere, I guess. If you don't have a standard sample where you know how it should look, and yeah, that would be my advice.
Great, thanks. Additional questions? So maybe you could just comment on your on your X-ray work, where do you expect to get to ultimately? Say 5 years from now, what kind of things do you expect to be seeing out of the X-ray work? We won't write it down. Go. You can just speak freely.
Well, if I say 5 years from now, it will probably be 10 because I expect it to be much further 5 years ago. But what we expect seeing in a couple of years is really these larger particles, the capsids, and then also those structures that have not been tackled in EM because they are low abundant. And in 5 years from now, I really would love to see the early assembly intermediates, because these are the most interesting to drug development. And if we get those, which are a few hundred kilodalton, and I would like to see them at, let's say, 3 to 4 angstrom, then we could also go for example, polymerase assemblies where we have heterogeneous samples, and let's say in 10 years, really go from heterogeneous mixtures, look at time resolved reactions and things like that. That's where I want to go.
That sounds great. Look forward to it. There is a question that came up in the chat from Weizen Kumaku. Excuse me if I mispronounced that. In slide 19, you showed the exchange of the GLU-77 into an AS78. Do you think that the exchange of acidic AA with another group can make the virus less virulent?
Could be. I mean, the question is then, does this change in the NSP78 complex affect the processivity of the polymerase and therefore the speed of replication, right? And if I think of the glutamate to asparagine exchange, then we also have in the virus the trimers coming up. So it could be that the replication is less efficient or that it's tuned to a different host. But to be honest, we don't know. What is certain is that the surface proteins do have an effect on pathogenicity, but there must be something intracellular as well. The spike alone is not doing the same. Can only speculate.
Thank you. Any additional questions at this time? Okay, if not, thank you, Charlotte. It's fantastic, interesting work. Look forward to hearing more about this. And thank you all for attending. As next month, we'll have Kyle Brown talking about his membrane protein work, and we'll have more information next week on that. So thank you again, Charlotte. We really appreciate it, and have a great day, everyone. Thank you. Goodbye.
You're welcome.
Bye-bye.